So thank you very much, Sebastian. First of all, I would like to express my sincere thanks to Sebastian for giving me this very honorable opportunity to have a talk at this very prestigious Collège de France. And actually, this is my first trip after COVID-19 pandemic to Europe. So my last trip to Europe maybe 5 or 6 years ago. So I'm really happy that I have an opportunity to come back to this beautiful country and nice opportunity to talk with my close, the friends face to face. Of course, we have some, sometimes communication through the Zoom, but the Zoom communication is. At the beginning, I found some interesting, but then it's become boring. So, so it's better. Face-to-face is much better than Zoom. So anyway, so as Sebastian told that now in the field of nanomedicine, many people are aware of how to translate their nanomedicine system into clinics. So in my talk today, maybe I would like to focus on our most recent effort to make nanomedicine, especially for delivery of oligonucleotides into clinical trial. Okay. Yeah. So So we have been working maybe more than 30 years on the self-assembly of block copolymers to form polymeric micelles. And of course, you can choose mainly, we are using polyethylene glycol as a hydrophilic segment. And of course, for core forming segment, of course, there are several, many choices. But as Seb told that we are mainly focusing on using polypeptides, that is, mainly by anionic polymerization of NCA. So because, as you may know, that polypeptides are biodegradable by and also biocompatible, and also you can introduce many functional groups in a side chain. So it's a very huge, wide versatility. And this, the micelle with polypeptide core can accommodate many different kind of, the cargo molecules, small molecular drugs, proteins, imaging reagents, and of course, the nucleic acid compounds. And our core is totally surrounded by dense polyethylene glycol layer to reduce non-specific interaction. And of course, you can, even conjugate some ligand to have active targeting properties. And a variety of interactions, bonding is applicable for drug loading, as shown here, the hydrophobic interaction, the coordinate bonding, and conditional covalent bonding. Most typical example is that, conjugating drugs through a pH-sensitive linker. And of course, ionic interaction is quite important for loading of proteins and also the nucleic acid compounds. And these, the systems loaded with mainly cytotoxic reagents, which we developed, have already been in clinical trial for, for treatment of cancer. But as Patrick, mentioned in his talk, we also encounter some difficulties for clinical trials. And for this one here, that is a cisplatin-loaded polymeric micelles have proceed into, finally, the phase 3 clinical trial for the treatment of pancreatic cancer, and by our established company, Nanocarrier. And as shown here, this is some part of the, the results. This is a domestic, the results in Japan. But actually, we had in combination therapy of gemcitabine and cisplatin micelles shows a significant, efficacy in medium overall survival compared to the, gemcitabine, treatment. However, it takes, long time for phase 3. It's same, yeah. So, and eventually what happened is that gemcitabine is not usually used for first-line treatment anymore because of the, the other, clinical modalities. So, unfortunately, the company, give up for the, commercialization of this system. So the point is that that's the reason why I asked you a question: how to, how to accelerate the, the late stage, clinical trial. Otherwise, even if your system, working very well, but always there is, competitors. And pharmaceutical companies often the case likes to have such kind of, new modalities than, utilizing the, the drug delivery formulation of already existing, cytotoxic reagents. So anyway, today I would like to focus more on our most recent, system, focusing on these, nucleic acid compounds. Of course, it's quite, itself has a very promising, modality, but and also as for the, clinical translation, now the pharmaceutical companies are very much interested in new modalities of nucleic acid compounds. So we have a more opportunity to collaborate, It's because, in cisplatin mice cells, our company itself, carried out to phase 3, but we learn and we become more smart. And so in this case, maybe we will, collaborate with already existing some pharmaceutical companies. So anyway, I would like to focus on this topic today. Okay. So of course, the already several, oligonucleotide drug formulation have already been approved. The first one is that, of course, some, oligonucleotides, mainly the antisense oligo, as approved as a free form. And because this is a structurally simple composition, that is the advantage. However, it's okay for the, local treatment. But if you inject this systemically, it lacks tissue specificity and exhibits a short circulation half-life. Mainly due to the renal clearance and recognition by so-called pattern recognition receptors, PRR, expressed in the whole endothelial surface of vasculatures. That is a very big problem, actually. So there are, several trends to conjugate some ligands directly to oligonucleotides. The typical one is GalNAc to target the acyl glycoprotein receptors, that expressed on the liver sinusoidal endothelial layer. And in this way, this is chemically simple and well-defined structure, and exhibits preferential accumulation in the liver by binding to acyl glycoprotein receptors, but still undergoes a non-specific clearance from circulation, same as a free form. So alternatively, not using this, the naked, form of oligonucleotides, there are of course several, the drug delivery formulations. The most typical example is a lipid nanoparticle, which become very successful for messenger RNA vaccine. So this, LNP has inherent, characteristics to predominantly accumulate in the liver. So this is, works well for liver targeting, and enables, endosomal escape, facilitating robust RNAi activity. But problem is, it's often the case, it's structurally complex formulation, and may trigger, that is a very big issue, may trigger lipid-induced inflammatory responses. And of course, this works, as an advantage in vaccine application, but it's become big, the disadvantage for other, applications, and still that it exhibits a short circulation half-life. So that means the extrahepatic delivery, especially a good example is solid tumor, is still challenging, the topic. So we are, actually challenging for oligonucleotide derivative system for extra hepatic region. So as for the solid tumor, everybody is now familiar with enhanced permeability and retention effect. The first three, the proposed by Professor Maeda and Dr. Matsumura a long time ago. So often the case, the EPR effect is, explained very simply. That is, the leaky vessel wall of, the solid tumors. And eventually, long circulating particles can have more opportunity to passively accumulate into solid tumors. But important point is that, in this Professor Maeda's, the original, the literature published in 1986, he doesn't use any nanoparticles. What he used is just proteins, and the albumin and IgG antibody. So actually, now everybody knows the antibody drugs are very successful for the tumor treatment, but actually these, the antibodies, including, the anti-PD-1 antibody, accumulate into solid tumor by some, the type of EPR effect. But the important point is that, of course, the translocate from blood compartment to tissue is the first step. But, the solid tumor is, mass. So you need the, effective penetration into tumor mass. That is another challenge. So actually, the many nanoparticle formulation, including ourselves, now move into clinical trial. But often the case, we encounter some problems, like a heterogeneity in EPR effect and stroma. So the clinical, solid tumors in clinical situation, usually it has a very thick stroma. So this, of course, become problem for this kind of tumor penetration. So this is the example of the pathological, histological section of human pancreatic cancer. As you see, that is very heterogeneous structures. So this, the dark blue region, that is a cancer cell nest. And green color is a vessel. And red is a stroma. So you can recognize that the vessel only exists inside of the stroma. There is no vessel directly in the cancer cell nest. So that means that even if your system can extravasate, still the needs to penetrate this thick stroma to reach to the final target, cancer cells. So that is a big problems. So how to, circumvent these problems? There may be three approaches. The one is the size modulation of nanosystems, make nanosystem size small, and use of reagents enhancing tumor permeability. That is the so-called utilizing some drugs to to change the vascular characteristics. And often the case, it has a tumor has a very strong solid solid pressure. So it collapses the vasculatures. So to utilize this reagent to circumvent these collapsed capillaries to work again. And of course, the use of ligands inducing active transport. So I would like to focus mainly to first topic, how size modulation, that is a very simple way, but how size modulation can circumvent these problems. So this is an old study about the size effect of nanoparticles, that penetrate into this stroma-rich pancreatic cancer. So you can see here that is the liposome with a size of 100 nanometers. You see all this 100 nanometer liposome just stuck in the vicinity of vasculatures and never get into the cancer cell nest. But in case of the good thing of the polymer therapeutics or polymer self-assembly is that by changing the polymer structure, you can easily tune the size in a wide range. So in this typical example, we prepared two types of micelles. one is 30 nanometer and another is 70 nanometer with very narrow distribution. So 30 nanometer micelles are, labeled with green fluorescence. 70 nanometer are labeled with red fluorescence. And we are observing the pancreatic cancer in, the subcutaneous, pancreatic cancer in mice by intravital confocal imaging. So that is the inside of the, living, cancer in living mouse. So you can see here, inside of the vessel, that is color is yellow, because that is a merged color of green and red. And this dark, the figures, these are the erythrocytes. So, but you see, inside of the pancreatic cancer, it's totally green. So that means only 30 nanometer mice can penetrate deeply into stroma-rich pancreatic cancer, but 70 nanometer mice all stuck in the vicinity of vessels. So size is quite important in terms of the penetration. So we designed the siRNA ASO-loaded nanosystems for enhanced tissue penetration, and also importantly is a streamlined formulation. The formulation should be easy to prepare. So as I mentioned, that compared to other type of the formulations, the polymeric micelle, we can downsizing the micelle to maybe 30 nanometers, but still the siRNA is around 4 nanometers. It's too small for because it shows a rapid the renal clearance. So the target region is here, in between below 40 nanometer, and size should be larger than the clearance limit of clearance limit of renal. So the target range may be corresponding to the size of antibody. And also, another important point is that there is a significant advance in chemical modification of oligonucleotides and have significantly improved their enzymatic stability and cellular permeability. So thereby reducing the need for carriers to provide protection against nucleus degradation or facilitate endosomal escape. So we decided to simplify the carrier design only with improved pharmacokinetic performance. So what we designed is so-called unit polyion complex. So you can see here, these oligonucleotides are of course polyanion. So just mixing this with the opposite charged block copolymer like polyethylene glycol polylysine, for example, then it spontaneously forms polyion complex micelles, and of course, this micelle works quite well for the delivery of, the protein and nucleic acids. However, important thing is that, the process is not, proceed in two ways. So first, by mixing this siRNA with opposite charged block polymer, what we can see is that the formation of unit PIC. That is the association of polymer with siRNA with compensating their charge in smallest, the association number. That is a unit PIC. Then this unit PIC, undergoes a secondary association to form polyion complex micelles. So we are focusing, of course, we are focusing this unit polyion complex, but usually, it undergoes a secondary association. So we try to stop this process, and also to improve the stealthiness to escape from scavengers in blood compartment. The answer is quite simple. So we just, elongate the PEG and make a two-arm system, and also make the charge of the polylysine in this case as half the number of the charge number of siRNA. So siRNA has a 40 charge, that means 20, and mixing this, it's a simple mixing. The results in the formation of unit polyion complex with a size almost comparable to antibody. So this is very, very simple system. So most of you concerned that how this kind of simple system can work in vivo. So I will show you how this works in vivo to stabilize, siRNA. So often the case, we utilize this intravitreal microscopy, by observing the thin capillary in the ear of animals. Of course, now there's a very strict regulation about animal study, especially in Europe. But the good thing of this kind of intravitreal microscopy is that you do not need to sacrifice animals. So, for example, in case of pharmacokinetics, that you have to sacrifice each time point for animal to take your blood or tissue. But by utilizing intravitreal microscopy, you do not need to sacrifice animals. So that is another advantage of this system. So okay, so we are now observing the inside of the capillaries and how to characterize the the integrity of siRNA. So we utilize fluorescence resonance energy transfer. So because the size of siRNA is around 5 nanometers. So we just conjugate each end of siRNA with donor and acceptor fluorescence dyes. So in case of the system, if you excite the donor, the energy transferred to acceptor, and you observe the very strong magenta color. But if siRNA undergoes the degradation, there should be no plate, so color should be blue. So left side is naked siRNA, and other side is siRNA loaded into unit PIC. So you can see the very big difference as shown here. So left side is immediately turned into blue, but right side it stays as a magenta. So actually, eventually we have the very longevity of intact siRNA under the blood circulation. and eventually we have a very, the high accumulation to tumor as shown here by this system. And also, in case of the pancreatic cancer, we observed under the microscopy the penetration into cancer cell nest. So in this case, we utilize, this is a BXPC3 human pancreatic cancer. It's a GFP system. So, each cancer cell, shine as a green fluorescence. And the red is the siRNA. So here is a stroma. It's totally dark. But you see in the cancer cell that you observe some, red spots that coming from siRNA, penetrate into cancer cell nest. And actually that is, the fluorescence intensity, the significant increase in the region of the tumor nest. And we are very lucky to have a collaboration with, clinicians in medical department, Professor Taniguchi and Professor Imai. So they are working on a tumor-specific transcription modulating factor named PRDM14. And as is shown here, this is a PRDM14 expression in human tissue, and this is white bar is normal, and blue bar is tumor. And always, most of the cancers, there is an overexpression of PRDM14, and especially the big difference in breast cancer and pancreatic cancer. So we load this PRDM14 into unit PIC and carry out animal study. And this is the efficacy against liver metastatic model of pancreatic cancer. In the control siRNA, there's a significant metastasis occurred in liver, but PRDM14, there's no such kind of the metastasis. And this is a survival curve. The upper curve is for the that is a triple negative, the breast cancer, and lower panel is that pancreatic cancer. And we had a very good result for the triple negative breast cancer. So then we tried next step should be translation to clinics. So before moving to, of course, to clinical trial, we have to ensure the safety of this block copolymers, especially the clearance, because you can see here the two-armed PEG total molecular weight 80K, very big. So it's larger than the renal clearance limit. So that means there should be no clearance, and then you cannot use this kind of polymer for never in clinical trials. But Anyway, we find out that this, the two-armed, block copolymer can smoothly, clearance through the liver. So again, we utilize, the intravital microscopy, continuously observing the clearance profile of this block copolymer from liver sinusoidal capillaries. So we inject the block copolymer, the labeled with red fluorescence. This is a free polymer. So after 1 hour, it's, the adsorb on the, surface of sinusoidal wall, but then 3 hour, 6 hour, 8 hour, it's gone and accumulate into the, this region. And actually, this region corresponding to bile. So this indicates that this polymer can smoothly excrete through the bile. And what about, adsorption to the other tissues? So we observe the, the behavior of this polymer in connective tissue. And you can see here, there's no adsorption to connective tissue. So that means that this polymer can, not adsorb on the other, endothelial layer of vasculature, except liver sinusoidal capillaries, and excrete from liver sinusoids. And this is, the more clear evidence of bile excretion, because this is a bile tracer green, and this is a polymer, and this is a margin. And you can see here that these polymers are all located with same region as bile tracer. So that means this polymer undergoes a transient adsorption to liver sinusoidal endothelial layer and excreted by a bile, so ensuring safety. And actually, it's, the smoothly excrete from blood circulation within 24 hours. And no critical side effects in non-human primates, and we established the GMP systems production. So we are now proceeding to phase 1 clinical trial of unit PIC SIPRDM14 system for treating recurrent breast cancer at the Cancer Institute Hospital in Tokyo. So maybe some of you are curious about why we need such kind of two-arm systems. The answer is that it's essential. So the reason is that, of course, we prepared also the single-armed, PEG block polymer with 80 kilodalton, but here is the, the adsorption to the sinusoidal capillary wall, but even after 9 hours, it's staying. So no excretion. No excretion. But as I show you, if you change to this two-arm, it smoothly excrete through the bile. So two-arm system is very important for the clearance. So anyway, there is a, phase 1 clinical trials has completed, and this is the, the IP from, our company, actually the nano mRNA. And you can see here the important point is that no severe adverse effects observed, indicating no major safety concern. And we obtain, this is just phase 1 clinical trial. So the main, the purpose is safety, the examination. But even in this case, we have a very long-term stable disease. And most importantly, from the, the pharmacokinetic or drug delivery, the standpoint is that the effective plasma concentration extrapolated from non-clinical results was achieved. So that means, the data obtained by animal experiments are, reconfirmed in human clinical trial. So that means that this system works as a very good drug delivery system with safety. So next challenge for this system is the treatment of glioblastoma. So see the glioblastoma is the most deadly form of human cancer, and median survival is only 10 to 14 months. It's really miserable. So again, we have a good collaboration with Professor Kondo at the medical school. He has been working on TAG1, that is a long non-coding RNA maintains malignant brain tumor stem cells. So if you knock down this, the tag 1 by siRNA, the human glioblastoma stem cell undergoes apoptosis. So based on this results, we collaborate with Professor Kondo to use our unit PI system for delivery of antisense oligo against tag 1. So take the glioblastoma stem cell from a human patient and inoculate again to the brain of the experimental animals and start a treatment with unit PIC loaded with antisense oligo targeting tag 1. So first we confirm the accumulation of tag 1 antisense oligo to glioblastoma, and then that is efficacy. Efficacy is very impressive. So you can see here that is a tumor volume. It's reduced more than 90%, almost gone. And that is a knockdown efficacy is of course more than 90%. And most interesting result is that we got 100% survival for 100 days for all the animals with the orthotopic brain glioblastoma. And eventually, the phase 1 clinical trial has started. It's is ongoing at Nagoya, Kyoto, and National Cancer Center hospitals. And this is still ongoing, but there is a IP from the progress report, and most importantly that the patient enrollment has progressed more smoothly than anticipated, and trial has now entered in third stage of phase 1, and next coming July, it will move into the final fourth stage of phase 1 clinical treatment. So of course the efficacy is not not opened yet, but we already have some the patient with long survivals. So looks very promising. So anyway, can I still have a time?
I think so.
Okay. Okay. So I would like to explain a little bit about the mechanism. So those who are working on the field of polyion complex, everybody knows, it's very difficult to separate polyanion, polycation. It's very strong interaction. However, if there is some amount of excess polymer, it's easy to exchange. So situation is more like this. That is some kind of country dance. Always you change the partner, but always you are dancing together. So point is that whether this kind of things occurs even in the body. So again, we utilize a FRET. So in this case, we label the polymer with donor fluorescence dye and siRNA with acceptor fluorescence dye. And first, we inject the free polymer into the mouse. So of course, in this case, of course, there should be no FRET. So color should be blue. And actually, it's blue. Then 5 minutes later, we inject a free siRNA. And if this polymer catch siRNA under the circulation, this color should be changed to magenta. And immediately after injection, it changed to magenta. So for the proof of concept, we inject half the amount of non-labeled polymer. So if it's exchanged, then the fluorescence intensity should be in between green. And it changed to green. So this result shows that clearly shows the circulating polymer can directly catch injected naked siRNA during circulation. So in French word, it's a French word rendezvous. So maybe my pronunciation is not so good. But anyway, we say rendezvous. So siRNA rendezvous with Pegasus polylysine in circulating blood, like a spaceship rendezvous in outer space. But outer space is somewhat easy because nothing. But in the blood, there are proteins, the cells, and so that is more like the moving crowd. So that is, even in this moving crowd, the person with red hat and red cap recognize each other and changing their caps. So that is the system ongoing in the body. So if I explain this in a different way, that is just like a rugby ball. So always there is a the enemy, and then pass the siRNA like this. So the unit PIC dynamically pair with circulating block cationers and capturing siRNA in the bloodstream, much like a pass in rugby. So this mechanism allows siRNA to be stabilized in transit. It's a dynamic stabilization and successfully score a try at the target site within the body. So the system is very, very simple, but anyway, it works. So finally, I would like to acknowledge all the, my colleagues and medical doctors in the hospitals, and of course, funding agencies. So thank you very much for your kind attention.